Okay, hi folks. So welcome back. I'm hoping that you all had a great summer vacation. So welcome to 360, where we're going to be talking about neurogenomics, so some of the genetic and molecular biological techniques that we can apply to understanding different aspects of behavior. And over the course of the next 11 weeks or so, we're going to be meeting here regularly, and then we're also going to be breaking up into smaller groups in tutorials in the last third of the semester. So not immediately, we'll be doing that a little bit later on. You'll be with a TA and you'll be with a small group that you'll be continuing on with, where you will have presentations with your TA throughout the rest of the course. So today I wanted to just give you a little bit of an overview. There is material because you know the first term test, and I hate to do this to you, but I want you to get some feedback early on. To be honest, I've been talking a lot over the course of orientation week to a number of different students, and I hadn't realized, and I went back and I actually double checked, I didn't like genetics. So it turns out that my first major was a major that's no longer offered here. It was called medical genetics. That was offered a long, long time ago, and Ashley was in medical genetics for all of, I think, 8 months. And then I decided that genetics wasn't for me. I switched out. I decided to go into farm talks, and then I decided that farm talks wasn't for me. And then I decided to go into physiology. But in the interim, actually genomics and genetics and molecular biology have actually become really exciting and very interesting. So I'm hoping you don't have sort of that same experience, that you're not going to get turned off of genetics, and that you'll understand the utility of it as we go through different aspects of today's lecture. So I am going to go through some of the biggest issues in neurogenomics. This is, by the way, being streamed, so if you miss something today, we do have an audio recording as well as having a screencast, so you'll get to see everything that I've done with audio sort of synced together. I would still like you to come to class as often as you can. Missing 1 or 2 is okay, but to be honest, you're going to be wasting about 3 or 4 more hours trying to listen to a lecture and take down notes, rather than being here for 2 hours and trying to understand things as we go along. So I'll give you a little bit of the course overview today, some of the different themes in the course, how I'm going to evaluate you. I will be uploading some more things related to the course on Thursday. So Thursdays are my administrative days. So what the tutorials will look like, what you should be doing to prepare to do really well in the course, I'll be uploading that for you on Thursday as well. Please don't feel shy about coming up and grabbing something to eat at any point in time. So if you're hungry, I'd rather you come up and grab something rather than sort of sitting there. Eating is a big part of life, and it's a big part of my course, so I hope that you will eat as much as you possibly can without overindulging yourself. So we are going to, in this course, talk a lot about different technologies as well as different techniques, applying them to a neuroscience background. Because it makes it more clinically relevant, we are going to talk about different diseases and their treatments, starting a little bit today and more next week on one of the most exciting things related to CRISPR-Cas9 technologies. And then we'll be looking at some of these emerging models. And in fact, all of these are going to be overlapping together. The fact that we can now grow stem cells, induce pluripotent stem cells, and then grow mini brains or mini organoids. Just to put things in context for you, in 2015, where some of you were just entering U of T or where you were thinking about coming to U of T, brain organoids first started to appear in journals, and it was one of the first sort of things that came out in Nature Neuroscience back in 2015. In a short 3 years later, we're actually at the stage now where in Toronto we actually have 3 labs that are working on brain organoids, and you'll hear a little bit more about that later on in the course. And these mini brains are actually revolutionizing the way that we think and we deal with how we treat diseases within the brain. So again, just to put things in perspective for you, and I want you to think a little bit about this. Over the last week, not just the summer, but over the last week, and this is just for context, I'll tell you when you should start maybe jotting down some notes. We had these really amazing papers. So CRISPR, which has been around since 2012 in mammalian systems, CRISPR has actually now been used to reverse some of the learning deficits and some of the behavioral deficits in an autism model in mice. To me, when I read that paper, it was really, really amazing to be able to go in in a fully mature mouse that has had symptoms and behavioral deficits that look a lot like autism in a mature setting, and to reverse them, that kind of goes contrary to everything that we learn about synaptic plasticity, the way that we understand behavior. And in these mice models, to be able to reverse that was one of those really spectacular papers that we'll be dealing with a little bit later on. You're also going to hear a lot in the newspaper, especially as we start the new school semester, about intelligence and other factors. And in fact, there was a paper that came out last week that actually suggested that your genetics actually determine whether or not you're going to succeed in university rather than sort of the environment. And these genes playing a greater role, those are really, really controversial as well. And I want you to be able to understand these papers that I'm going to be sharing with you, and as we'll sort of unwrap them. So as we go through these GWAS studies, how can we make sense of it? Is this actually a good paper? Is this sort of a sensationalist paper? Even if it's published in something like PNAS or something like Nature Neuroscience, does that necessarily mean that the science behind it is all that sound? So this is sort of a minor reboot. We've changed some of the topics, and we want to continue to change things and modify them. So it's a little bit of a minor reboot from HMB360. There are some exciting things that we're going to try this year, and I'll go through that with you a little bit later on. And I want you to understand that this is a field that most likely will lead to sort of the next Nobel Prize in physiology or medicine, in our discipline of genetics, or in neuroscience. This is really the nexus of where exciting things are happening in the field of biology, and I want you to feel that same excitement. This is really an amazing place to be in right now. So again, a little bit about the course. Most weeks, we're only going to meet for 2 hours. So even though in your schedule it says that we meet from 10 o'clock to 1 o'clock, we'll really have lectures most weeks from 10 to 12 o'clock. That extra hour, 12 to 1, we're really only going to use that toward the last third of the course in November, where you're going to have that 12 to 1 hour for presentations. So you don't need to worry that you have to be here for 3 hours. To be honest, I wouldn't want to lecture for 3 hours, nor do I think you'd want to hear me lecture for 3 hours. So just think of it sort of as a 2-hour lecture. You will have a lot of group work in this course. And I know some of you are very, very anti-group work, that you don't like it, you don't appreciate it. And I want you to refocus a little bit on why group work might be really important. So I want you to be thinking not only the presentation of a paper that you choose together as a group to your TA and to your peers, but also we're going to be working on developing a textbook together in this course. It will be published, it will be online. People will be checking it out, and I want it to be of sufficient quality that you feel very proud of the work that you have, and not just get a good mark from your TA, but also understand that you've created something that's going to be sort of an online textbook that other people will be using, including next year's class. So I want you to be very excited by that. Wherever possible, we're also going to have reviews before tests and exams. We'll also make sure that all of the things that you need to know to do well on these assignments, we have some kind of in-class activity to get you prepared ahead of time. So we do want to cover topics that are relevant for you in neurogenomics, as well as in neuroscience. And as students within HMB, we want you to get really excited by these different types of things that we'll be looking at. So as I mentioned, we're going to be looking at a lot of different technologies, different applications in neurogenomics. These are always evolving. If you could pinpoint one paper, so the one paper, especially if you're a neuroscience student, what was that one paper that came out in the summer that kind of took everyone by surprise, that was really different, that no one had really expected? Any ideas? Any guesses? Like I know all you did all summer was read papers, so share a couple of them with me. What was that one really groundbreaking paper? Was on every social media feed, everyone got really excited by it. What was that one paper? Any ideas? Any guesses? Anything that you read? Was the Cardi B neuron and what it was doing? Or the Cardi B pathway? Not that one, but maybe something else? Anything? Are you incredibly shy? No? You do read papers though, right? Ooh, a lot of nervous laughter. That doesn't sound good. So maybe the one paper that was really surprising was the identification of a brand new type of neuron within the human brain. This neuron that has been there since our brains have evolved is called the rosehip neuron. And one of the things that we're going to be looking at, this idea of using single cell RNA and being able to identify neurons that we never suspected existed before is actually a really big part of both genomics as well as neuroscience. So we'll be looking at all of this, including organoids and how organoids help us in understanding a number of these different neurodegenerative conditions, as well as conditions that are more developmental, things like autism spectrum disorder, and how these brain organoids tell us about proper connectivity as well as proper functioning in our field. These are also very important in terms of ethics. We now think that these ethical issues that we never thought about before, and we'll get to this sort of in week 10. So if you humanize a mouse, which is often done in our disciplines, so you take human cells and you transplant them into a mouse model or a mouse brain, what are the ethics of that? And we never used to have to think about that before, but now it's becoming a really big issue in terms of the ethics behind humanizing different types of animal models. So we'll be covering the gamut of all of these different types of things. So again, in terms of what this course is going to look like for you, there's going to be some tutorials, and we have one of your TAs here. I don't want to keep Clara too long, so Clara, if you want to just stick up your hand. So about a third of you will be with Clara. Victoria couldn't make it this morning, but you'll have Victoria as well as your third TA, Justin, who is also in neurogenomics. So you'll be with them throughout the semester. They'll tell you like how to do well on your presentations, as well as how to do well on the different types of assignments. So I will, for example, give you different types of review sessions before the midterm, final exam, etc. So please make sure that you're coming to class, checking on Quarkus as much as you can. I know there's a big temptation not to do so. Try to log on at least twice a week, at the beginning of the week, end of the week. If you can't do that every day, which I understand, at least check in twice a week if you at all can, because I do tend to use it an awful lot. So here's where you get an opportunity to tell me a little bit about how we might be able to change things. So if you looked at the course syllabus before coming to class, you'll know that this is sort of the overall breakdown. And I want you to understand that there's some flexible options. Some of you really like writing term tests. How many of you love term tests? Okay. How many of you love presentations, assignments, writing? Okay, so I don't know about the rest of you who didn't put up your hand. I'm sure you like something. We'll figure that out as we go along. But I know that sometimes, for a variety of reasons, students don't do quite as well on a term test as they had hoped, and then they kind of want to have some kind of other assignment, etc., to kind of bring their marks back up. So we want to give you that flexibility that whatever you do better on, that it will be weighted in your favor. So if your midterm was spectacular, you had 100%, you didn't do so well because things caught up to you at the end of the semester and you didn't do quite as well on your final assignment, then maybe it's going to be worth a little bit less than your first term test, and we'll use that flexibility option there. The other thing, how many of you, if you like term tests, how many of you love final exams? Writing final exams are my favorite thing ever. Like, I look forward to them all September, all October, November, and when December gets around, like, you get all excited. That final exam, really looking forward to it. So I know some of you get very, very uncomfortable with the final exam. So in advance of the final exam, which technically is 30%, we are going to have some online quizzes. We will use 5 of those questions from those online quizzes toward the final exam. In addition to that, every quiz that you write will reduce the weight of the final exam by up to 1%. So if you write 5, you will have a final exam that is only worth 25%. If you can't write all 5, you're not going to be penalized. If you write 1, that means that your final exam will just be worth 29%, and you'll have that 1% sort of tacked on to it. So I'll again explain that to you a little bit more as we get further along in the course, but I wanted you to understand there's a lot of flexibility in this course. It depends a lot on what you do throughout this course as well. Please make sure you're staying on top of things as much as you possibly can. So again, these extra presentation times will take place on the same days, on Mondays, and this will be from 12 to 1 o'clock. Or 10 after 12 to 1 o'clock. We're going to try to find rooms that are actually very close to where we have lectures, so you don't have to worry about walking across to other parts of campus, which will split up into different rooms. There will be about a third of you in each of the rooms, and you'll be doing your presentations. This will be sort of in November, in the last 3 weeks or so of November, and you'll be presenting on these alternating weeks throughout the course of November to the rest of your small group. And you'll be with one TA. That TA will be responsible also for marking your final sort of chapter that you're writing based on the paper that you've chosen to present. You'll be expanding on it, and you will be working together as a group on that sort of chapter. I'll give you specific things to include in that chapter, and as long as you meet those requirements at a sufficient level, then you should be able to get a really good mark on that final multimedia assignment. The only other time I need you to really be around between 12 and 1 o'clock is that after the first term test, we are going to have a group activity with everyone. So you don't have to be in the same group. We're going to be rewriting the same term test again as a group. That will count as your mark. Usually the average on the group term test, which will be a small percentage of your participation, usually it's pretty close to a 99%. So I'm hoping that you guys will stick around for that. It tells you like how you've done on your test almost immediately. You get to compare answers with your group mates, and you'll write that group, submit that term test again as a group, and that's sort of mandatory. I want everyone in this class to get to know each other as much as possible, and there will be one workshop, and just one workshop, where we go through what you need to do for your textbook. It won't be a full hour, it'll be about 30 minutes, but those two are going to be mandatory because I'd like everyone to do well. So here's sort of my rationale for why I'm hoping that you will be okay with group work. To be honest, I had some really bad experiences with group work myself, where I've had individuals not show up, not do things, tell me that they were too busy to actually complete their part of the assignment. I've had people quit halfway through an assignment, and this is like after I graduated, so these things happen. But I want you to understand that when I'm asked to write a reference letter for you, and I'm hoping that I will be able to write one for you, that when I'm asked about your ability to work in a group, so number 10, for example, your ability to be able to organize the efforts of other individuals, your ability to work with other people, number 9, these are very common now in reference letters. I can't do that with all honesty unless I've actually seen you work in a group or heard from your TA that you work really well in a group. And this is increasingly asked of in grad school, whether you're applying to professional school, if you're thinking about OT, PT, nursing, med school, etc. These are the types of questions that get asked, and I'd like to be able to write a complete reference letter for everyone in class, and the only way I can do that is if I know that you can work well in a group, either for point 9 or point 10. So again, that's going to be your choice. You're going to pick the groups. I'm not going to choose them for you. Again, it's a contentious issue. Should we choose and make you go into groups that you're not comfortable with, or should you be allowed to find other individuals that you're comfortable working with? And these will be small groups, somewhere between 3 and 4 students. If it's a little bigger or a little bit smaller, that's fine. You're going to be finding a primary paper, which is very bothersome to me because I'm not sure if you read papers or not. I hope that you do, so I'm going to be posting as many of these as I can. You're free to choose any of these papers as long as within that group that you're in, whatever TA that you're with, that paper is not covered by someone else. And you're going to pick something that is of interest to you. The other thing that's really important, don't just pick a paper that interests you, also be able to understand it, which means that if you're choosing a paper a week before the assignment is due, you haven't really read it, you're not going to do really well. if you pick a paper that interests you and you spend time over like 2 or 3 weeks, you come and see me or you talk to your TA, you're going to do so much better on it. And I can guarantee you that everyone who did really well last year did exactly that. They chose a paper early and they decided this isn't a paper that's going to work for our group because none of us understand it and none of us actually felt that we could present it, or that you're going to come back and do other types of things, or other papers. So I am going to give you some guidelines for this starting this Thursday. You'll get sort of the assignment guidelines so you can map out what you need to do throughout this semester. I want you to be really well prepared. And again, please check online as frequently as you can. We'll map out and give you examples for everything. As long as you organize your time well in this course, you can get an A or higher. It's not going to be a course where it's going to be punitive for you. We are going to, as a class, so we're hoping that as a class, you're going to have somewhere around 25 to 30 different chapters that will be put together individually or as these groups into one textbook that is going to be published online, and eCampus Ontario has agreed to publish it both in BC as well as in Ontario, so you will actually be a published author by the end of this course. And as I said, you will have one workshop, and just one, about 30 minutes long, on what you need to include in the chapter that you're going to have. And there will be some more instructions on this as we go along. And again, I'll also put some of these links up there for you. Last year's project was a little different, but it gives you a good guideline for what you need to do. The more interactive it is, the more sorts of things that you can make your textbook interesting, the better your mark will be, but there are some caveats to that as well. So I've just done a whole bunch of talking over the last 20 minutes or so. Do you have any questions on anything that I can clear up for you? Something that I might have said earlier, something that wasn't so clear? Is there anything I can clear up for you now? Yeah, any types of questions? Is it going to be like this all semester? Please don't make it like this all semester. I'd really love it if you ask questions. It's not that big a class. I'd like you to feel comfortable enough to ask questions as well. So I'm hoping that you're going to come to class every single week. I'd like you to stay on top of the material as much as you possibly can. Some of your classmates and I are going to be working to give you a weekly study guide. I'm not going to write it myself, but I'm going to be working with some of your classmates. If you're interested in helping with that, please let me know. We're always looking to have more individuals join. And again, my suggestion to you, if you are planning to skip classes because you have another term test the same day, or you have another big assignment, missing 1 or 2 classes, to be honest, is sort of the U of T experience. You've all done it. I've done it. In fact, I was in a course where I didn't show up to a single class. And that was the course where I didn't do as badly as I thought. And I think that it's your choice, but I'm going to also tell you, you're going to spend more time re-listening to the lecture recording that's going to be provided than actually sitting in here for 2 hours. So use that as a resource, but don't use it as a reason for skipping. Try to show up to class as often as you can. It may sound like it's a really trivial thing to do, but I can guarantee you that it will make a difference by the end of the semester. Because the one thing that I don't want you to be thinking of, and this turns out to be a real place, are places like this. And for those of you who have checked out sort of things on social media, there is actually a disappointment island, there's a disappointment lake. These are real places. U of T is really great at sending you there. It's a one-way trip though. There's no return trip. You go there and you get really disappointed, and unfortunately, if you're visiting like Sorrow Islands, there is no way back. So you don't want to be visiting there, and I want you to be staying on top of things as much as possible throughout the semester. Our goal as a team with Clara, with Justin, as well as with Victoria is to make sure you're well prepared for every assignment, for every term test, every week. But it's your sort of responsibility to make sure that you're making use of all of these different resources. Do you have any questions or anything that you'd like to suggest that we do in this course? Yes, Ellis.
The paper we have to choose for our group work, does it have to do with neurogenomics?
It has to deal with either a genetic and brain disorder or something related to the nervous system, or it has to be related to a molecular technique and the nervous system. But you're pretty open outside of that. Anything else that I can answer for you now? Any other suggestions? Is there something that I haven't thought of that you would like to have in this course? Nothing. Yes, at the back, please.
So I was checking with the links that you provide on the the slide. And
Yes.
I just want to ask, how multimedia do you expect?
Yeah, so I'm going to give you, thank you for the question. I'm going to give you an assignment guideline as well as the rubric ahead of time so that you can start thinking about the assignment. It's not going to be completely multimedia. You're still going to have to do some writing because it is a textbook. But most modern textbooks now aren't like the ones that you buy from the bookstore. In fact, there are going to be specific guidelines where you should be asking, like if you've read, there should be some kind of way to determine if someone understands everything that you've just written. But there should be some links or pictures that you find. You should be citing them. You should be making sure that those are all labeled for you. You don't have to go out and shoot your own video. If you want to, fine, that's great. If you are not comfortable with drawing something by yourself, we can help you with that. There's also going to be access to sort of a multimedia suite called BioRender for everyone in class. So you can easily design sort of your own pathway. We want you to be creating something that you're proud of. So I don't want you to think of it as a stressful thing. So it doesn't all have to be multimedia. There's going to be writing, and it's going to be about 50-50. So when you're discussing that as a group, some of you might be really artistically inclined, some of you might be better at researching, some of you might be better at writing. It's up to you how you design your group. Any other questions that I can answer for you? Yes?
Will we be receiving one mark as a group or individual marks?
So 80% of your mark will be as a group, and then there'll be sort of 20% where you'll also indicate what you've done individually, and you'll be graded on that. Okay? So yes, there's always sort of a group and then an individual component. Part of that is because a few years back, there were some complaints that some students actually didn't deserve to get the full group mark because they really didn't do anything. And some students also, we felt, they did much more than what they had indicated for their group, and the TA was aware of that, so we wanted to make sure it's fair for everyone. Generally, most of the time, the group mark is fairly reflective of everyone's contributions, but we do also want to acknowledge that maybe for whatever reasons someone didn't do as their own job, or you did a little extra. Because U of T is all about being extra, right? Any other questions? No? Okay, so if you don't mind, I may just spend about 10 minutes, then we'll take a break, and then we'll sort of wrap things up. I don't want it to be too heavy, but I do actually want you to be aware that your first term test is in 3 weeks. So it's a terrible thing, but I want you to be well prepared for it. So this paper that came out from Eric Olson's group, and you don't ever have to worry about knowing the title of an article or anything else, but you're going to be hearing a lot about Eric Olson. He's not here, he's the senior author. This paper was actually one that came out not too long ago, about 10 days ago. And I want you to be aware that right now, in terms of gene editing, this is the most exciting time. You have access to this incredible technique. Some of you who are taking HMB360, where you're in the lab, you'll actually be working with CRISPR-Cas9 technology. So as an undergrad lab, to be able to do that, design your own sort of guide RNAs and actually do some gene editing. It's pretty amazing to me for you to be able to do that. But here's where we need you to start thinking a little bit. So this paper, which was a high-profile paper that made headlines everywhere, that was published in Science by Eric Olson's group, actually isn't all that different from the 2015 paper that came out that I'm going to share with you in a few moments, and then next week we're going to come and cover this. So how many of you have heard of a mouse model in genetics? So surprisingly, not everyone's putting up their hands. That's okay. Most of the time we've used mice. Here, we're actually looking at a canine model, and this actually is different enough. Why do you think a canine model for Duchenne's muscular dystrophy is important? Like, you don't get to publish in Science for just doing the same experiment over and over again. Here, they changed their model. They went and did this originally in 2015 in mice. Now, in 2018, they are publishing in canine models. What makes it different? Any ideas? Is it all that big in terms of the news? Yes?
That if there's a commonality between the mouse and the canine model.
Okay, so commonality between the mouse model, canine model, sure, there is some commonality, yes?
Maybe just because like dogs are bigger and their muscular system is more like developed, more like humans.
Okay, great answer. Anything else that you can think of off the top of your head? Because I want you to think in this course, like why is it published in Science? Anything else? How many of you have, yes?
Maybe the canine model is closer to the human version.
Okay, so maybe there's a difference between the canine versus the mouse model and it's closer to the human system. And yes, did you have another idea?
It was sort of similar to hers that dogs are bigger, so maybe we'll be able to see things that you wouldn't be able to see in mice.
Okay, so they're bigger, maybe we might be able to see other things that are going on in the canine model. In fact, all of these answers are kind of right because one of the reasons why Eric Olson decided to use this canine model 3 years later is that there is a new canine model that was developed, much more similar than the models that we're going to be looking at, the MDX mouse model that you should know for your term test, and we're going to get to this. And the canine model is exactly as you've described. The animal itself is much bigger, so closer in size to the human. You need to know, is this technique going to work in bigger tissue or bigger muscles? And the dog is much closer, much bigger in size. The dog also lives longer, so you can't really tell if something is going to be long-lasting in a mouse, which has a life expectancy of about 2 years, and dogs have a much longer life expectancy. Are these effects actually long-term effects? And I want you to be thinking as we go throughout this course, like why are they using these particular models? Are they good models, etc.? And we're going to come back and circle back to this in next week's class. So I need you to revisit, in some cases, a little bit about muscular dystrophy. It's a very, very common disorder. Muscular dystrophy, or wasting of the muscles, is actually much more common than we give it credit for. All of the muscular dystrophies that we see are all genetic in origin. So these are inherited disorders. Sometimes they're the result of spontaneous mutations, but whatever it is, these are genetic in origin. And we can always trace them back to a specific set of genes. And these are progressive. So once you have been diagnosed with this, there's no way to stop it from getting worse. These are progressive disorders in that sense, and you get severe muscle weakness. Now, the different types of muscular dystrophy lead to different types of muscle weakness, which may or may not be fatal. So we often talk about Duchenne's muscular dystrophy, probably the most common. In fact, 50% of muscular dystrophies are the Duchenne type, but in fact, every single type that you look at will eventually lead to a loss of muscle tissue. In your generation, one of the things that is the most important aspect in aging is actually the loss of muscle tissue. It is not something that we used to think very much about, that when you get older, there is a natural decline known as sarcopenia in your muscle mass. And it used to be, well, it's going to happen, so why not let it happen to an older individual? But we now think that that loss of muscle tissue as you get older actually directly contributes to the loss of health status in you. So in every course that you take, you'll hear about exercise, whether it's a aerobic form of exercise or things that will increase muscle mass. These are now thought to be really important in the aging population. So we know that in different types of inherited muscular dystrophy, we can see the effects of this muscle wasting or muscle weakness either in childhood, these are very, very severe, or in adulthood, less severe forms of muscular dystrophy. And some of these different symptoms, depending on when they occur, can lead to a number of different effects. So they can affect one muscle group at a time. They can affect several muscle groups at a time. So muscle groups meaning like your skeletal muscles within your biceps that control like how you physically contract your arm, or the ones in your legs, or the ones in your eye, or the ones in your back. These are different muscle groups, and how they get affected and the timing in which they get affected is going to be really important. So depending on the muscle groups that get affected, you might have delayed motor skills, you may have loss of limb usage, you might have a droopiness, which we call ptosis. So oftentimes you'll see sort of a muscle group that's no longer able to contract, and you'll see sort of a drooping eyelid or a drooping face. And this ptosis is actually one of the signs that your muscles are no longer able to contract in a coordinated fashion. And of course, ultimately, as your muscles stop contracting, they will lose size. And as they lose size, you're going to lose strength associated with these muscles. That happens in aging, but imagine having this when you are young, or imagine having this when you should be developing your muscle mass as you get a little bit older. So we'll go through this one slide and then we'll take a break if that works for everyone. So the two forms that I'm going to be talking to you about the most over the next lecture and a half are Duchenne's muscular dystrophy, which I'm going to abbreviate as DMD, and Becker's muscular dystrophy. These are sort of related. Do you have a question? Were you just stretching?
Yeah.
We'll stretch as a class in about 2 minutes. So if we take a look at these two, and they're the ones that we're going to be concentrating on, these are the ones that I'd like you to study for the term test. Again, if you're really interested in it, there are a number of other types of muscular dystrophy. You are not required to know all of them. This is not a clinical class. This is a genomics class. But there are myotonic forms, which are, again, later onset, adult onset. Usually they involve muscle groups in the face as well as neck, so you won't be able to use them. Your speech may be affected in myotonic forms of muscular dystrophy. There is the FSHD type of muscular dystrophy as well, also occurring more toward the end of the teenage period, early adulthood, affecting muscle groups within the face as well as in your shoulder. And then the limb girdle type. And this limb girdle type is actually a subset of the Duchenne's muscular dystrophy. And again, when you have these lower limbs being affected, this does affect your motor development. It does affect some of the larger groups of muscles in your legs. These are your biggest ones that are skeletal in nature. So the limb girdle type are kind of a subset of this muscular dystrophy type that we'll be seeing a little bit later on. We have a number of other types, all genetic in origin, all affecting sort of the same locus on the X chromosome that we'll get to after the break. So I think now is sort of a good time to take a break. I know you guys are just coming back from summer vacation, so it's a little hard to get back in the swing of things. But if you want, we'll sort of pick things up at about 11:05, so in about 15 minutes or so, and then I'll cover the rest of the slides today. Please feel free to help yourselves. Talk to Clara if you want, if you're interested in what her research is. She'll tell you in the tutorial herself. But again, just take a good break, do some stretching. Washrooms are outside around the corner, and help yourself to some food. Okay, folks, if you don't mind, we'll just get going, and then we'll try to finish a little bit early today. I know some of you have to get back to the other side of campus, or if you have any questions, I'll be happy to sort of stick around. The 12 to 1 slot in terms of the time, usually I'll use that for an extra office hour if you have any other questions. And there were some great suggestions, by the way. Please keep them coming. Some students already have an idea of the topic, maybe not necessarily the paper. So we will be opening up the discussion board where you can indicate whether or not you would like to join the group. If someone is doing a disorder or a disease that you're particularly interested in, you can always sign up and indicate that you'd like to be part of that group, which I thought was an excellent suggestion. Today's lecture recording will be posted for you sometime by the end of the day, I'm hoping, and you can let me know if it works for you as well. So as I mentioned to you just before the break, there are a number of different subtypes of Duchenne's muscular dystrophy or Beck's muscular dystrophy. We're going to be concentrating on DMD and BMD, or Beck's muscular dystrophy, or Becker's types of muscular dystrophy. And these are the muscle groups that are specifically targeted by these disorders. And again, the muscle groups are always different in the different forms of muscular dystrophy. These are the other types that we just talked a little bit about. You can see that the limb girdle type, as I've indicated for you earlier, is sort of a subtype affecting the same groups of muscles that we often see in Duchenne's and Becker's muscular dystrophy. And these are just ones that you need to be thinking about. I don't need you to memorize this slide at all. This is a non-important slide, as is indicated for you. So for Duchenne's and for Becker's muscular dystrophy, these are X-linked recessive conditions. So males are affected, females are not. Females may have some of the phenotypes, but generally they don't have the muscle dystrophy or the wasting of muscle tissue. They will have other sort of signs and symptoms, but it is males that will have this. It is X-linked, it is on the X chromosome. And so typically we would see this in a son that would have Duchenne's muscular dystrophy. They may look normal when they're born. They may have all of the normal developmental milestones, but then they might start actually regressing as they get a little bit older. So again, both Duchenne's and Becker's muscular dystrophy are affecting boys, female carriers, because the X chromosome is affected. They may have milder symptoms or sometimes no symptoms at all. For Duchenne's muscular dystrophy, the onset of the muscle wasting of those muscle groups, especially upper arms, lower legs, is typically early on in development. And it is much more rapid. And unfortunately, we have no cure for this. It is a fairly common disorder. It is a fairly common genetic disorder. And one of the reasons why so many researchers are so interested in this is because it is a fatal disorder, unfortunately. It is progressive, which means that once you have this diagnosis, so the child that used to be able to crawl around, stand up, when they turn 3 years old, they may no longer have that coordination anymore, their muscles are getting weaker and weaker. Unfortunately for them, there is no good diagnosis. So up to a third, by the way, of individuals that are diagnosed with Duchenne's muscular dystrophy, and the numbers are actually very controversial. So the sort of biggest group, about a third. Some people think that it's maybe as small as 1%. So there's a lot of variation, but up to a third of individuals have been suspected of having spontaneous mutations in the gene that I'm going to describe for you next. And in terms of the signs and symptoms, these typically appear when the young male is already learned how to walk, so everything looks normal. There is no reason to suspect anything is going to go wrong. And then, after they've started learning how to walk, things don't turn out so well for them. And then they get that muscle wasting, the lack of coordination, and then this is, again, somewhere around 2 to 3 years of age. And unfortunately, they only have a lifespan of about 10 to 12 years, and they will typically die in their teenage years. Their biceps muscle, for example, will be much, much smaller. It will start to degenerate and disappear, which is what this small diagram is showing you here. So again, this is a slide that you might want to spend a little bit of time on, but only in the way that I'm going to describe it for you. So as you'll see, I have slides that have both bullet points, and the bullet points are generally there for you to go back and study. Then I have slides that have nothing but pictures on them. So this slide here, for example, is a good one where you should be taking notes, whether or not it's something that you need to know or you don't need to know, or some extra details that I'll be providing for you. So one of the things that we think, and one of the most common forms of Duchenne's muscular dystrophy, if we're looking at the X chromosome, and the X chromosome having both the long arm and the short arm being the P arm, if we're looking at the 21 band or the P21 locus on the X chromosome, this is usually where we find some kind of mutation associated with Duchenne's. This is where we normally will form the dystrophin protein, and this is where we see a lot of these different types of mutations. And one of the things that we will often see on the X chromosome associated with Duchenne's is we will have a deletion of a number of these different putative exons, a fair number of them. And in fact, one of the things that happens is that because there is a large deletion of these different exons, the numbers you do not have to memorize. I'm not going to test you on, is it exon 45 to 54, etc. for Duchenne's? You do not. But I do need you to know that there are deletions of a number of these exons within the X chromosome that will lead to an out-of-frame dystrophin transcript being formed. And what happens is that there is a premature stop codon that gets introduced. So here you are with this gene. We now have these deletions, and instead of forming this really large protein, you get a smaller truncated version of this protein, dystrophin, that I'm going to explain in some more detail, meaning that you don't have a fully working version of the dystrophin gene or the protein. It's very similar for Becker's as well, and one of the things that happens in Becker's, we have a dystrophin protein that results in a somewhat functional and somewhat more functional version of dystrophin, but it is not fully complete either. And again, this sort of indicates the level of completeness. In Duchenne's muscular dystrophy, it's quite a big truncation, and in Becker's, it's a modest but significant change in dystrophin as well. So on this side of the diagram, at this time, you don't really need to worry about what's here. I'm going to go through this on a different diagram with you in terms of what the structure of dystrophin looks like, and this is just indicating the structure. But on this particular diagram, I don't need you to worry too much about it. So again, this is another slide where I'm introducing pictures to you, but here, the only thing that you should be taking away is that one of the clinical features of Duchenne's muscular dystrophy, so if I were to ask you, if someone shows Gower's sign, I need you to immediately link that with a form of muscular dystrophy. So we will, by the time your term test comes around, have covered a number of different types of disorders, and Gower's sign is one clinical sign that is very often present in Duchenne's muscular dystrophy. So here is a young individual, and you can tell that this slide was from a number of years ago before sort of the ethics in being able to identify a particular individual. So nowadays we wouldn't be able to show you their face, but you can see that for Gower's sign, they're not able to stand up by themselves anymore. And remember, they have been developing normally, and this is an older child, I think about 5 years of age. They should be able to stand up, walk around by themselves, but because the muscle groups in their arms and legs have been starting to get smaller, they've been dystrophic, one of the things that happens is they can't stand up like you normally would. They actually have to push themselves off, and they stand up in a very characteristic way where they might turn themselves over, and then they will literally walk themselves upwards. So they will start, as you see here on the right, they will start to walk themselves, and they will not just walk themselves to a standing position. Because their upper legs are so weak, they will actually walk themselves up their own body. And this is what Gower's sign is, which is what you are seeing here. They can't really do the things that they were able to do before. A very characteristic clinical sign, because those specific muscle groups that I showed you earlier, they are in decline. And they can't really have that posture development that they had before. So this hip muscle atrophy, the upper legs being atrophied, then the muscle groups being atrophied lead to this Gower sign as a form of compensation. Your muscles, or their muscles, are under attack. Their muscles are degenerating. In fact, every time they contract their muscle, another group of muscle degenerates and dies. And so one of the things that we often see, and hopefully you have all taken physiology, correct? You guys all did brilliantly in physiology, correct? Yeah. Okay, that's okay. I didn't either. That was my major, so I have to say that I did fairly well in it, but I didn't actually. So one of the things that we have within muscle tissue is this creatine phosphokinase. It's involved in sort of the regeneration of ATP. And one of the things that happens is that when your muscle degenerates, this enzyme that is normally found in intact muscle tissue, as that muscle degenerates, this gets released. We normally don't see this unless you've been working out really, really strenuously or you've had a heart attack, etc. And in fact, the one thing that we look for is a particular isoform in Duchenne's muscular dystrophy, which is known as creatine phosphokinase, or CPK-MM. If we see that, we know it's from skeletal muscle. It's not from damaged heart tissue, it's your skeletal muscle that is breaking down. The more we see, the faster your muscle is degenerating. And in Duchenne's muscular dystrophy, we would be able to see this quite a lot. The next step would be to actually go in and see if there is loss because you can lose muscle tissue for a number of reasons. It could be parasitic, it could be for infectious reasons, etc., is to go in and take a muscle biopsy. And when you take a little piece of skeletal muscle from an area that you suspect might be affected, you would actually see either a decrease substantially compared to their age range, or an absence of this protein called muscle dystrophin. So we just looked at the gene for dystrophin. We know it's truncated, and we are going to lose this in Duchenne's muscular dystrophy. There are also now genetic tests to look for any type of X-linked abnormality in the short arm of the chromosome as well, particularly in band 21. So there are genetic tests that could also be done to make this diagnosis for Duchenne's muscular dystrophy. This is not only for skeletal muscle. Dystrophin is also used by other types of muscle tissue, including cardiac muscle tissue. So not only do we have problems with skeletal muscle in Duchenne's muscular dystrophy, but we also have problems with cardiac muscle, and this will often result in arrhythmias, it will result in cardiomyopathy. If this goes on long enough because the heart muscle degenerates, you will have what is known as congestive heart failure. The heart muscle cannot pump as effectively as it should normally, and so the blood starts backfilling in the cardiovascular system, and you get what's known as CHF, or congenital heart failure. So this muscle mass, again, in the heart, in your skeletal muscles, will continue to decrease as this disease progresses. Again, in terms of Duchenne's muscular dystrophy, and again, another thing to make note of. Some of you have had me in other courses. For some of you, it's the first time that you've had a course with me, or it might have been a few years. I will point out generally what you need to know here. In terms of the size, you don't need to worry about the details that are down here on the bottom, but it's just to indicate for you on the bottom exactly what I pointed out for you here at the top. That if you were looking at this gene, this is actually one of the largest genes. So there's about 2.5 mega base pairs. Here it's indicated for you as about 2,500 kilobase pairs, which is the same. And this encodes for a fairly large protein. Again, the number not being important. I'll tell you if you ever have to memorize a number. I'm not big on memorizing numbers, but this is a fairly large protein. For those of you who do Western blots or do research, 427 kilodaltons, it wouldn't migrate very far in a standard protein gel because it is so massive. And so this is a large protein that we'll get to in a moment. This is found again in skeletal muscle, no surprise there, cardiac muscle. In fact, we do see dystrophin in non-contractile tissue as well, and I'll get to that in the next lecture. So it is indeed in other tissue types as well. And the reason why the loss of this protein is so important, when you look at muscle, it's typically actin and myosin. And when you look at these two different thick and thin fibers that cause muscles to contract, whether in the heart or whether in skeletal muscle, the link between the actin and the extracellular matrix is made through this dystrophin complex. And without this dystrophin, every time your muscle contracts, it is going to cause the muscle to tear, and every time it tears, there's a possibility that there will be an accumulation or an abnormal accumulation of things like calcium. Again, this is not a diagram that I need you to memorize every aspect of. And I'm not going to ask you to draw this at all, but I wanted you to see this based on the last slide that I mentioned to you. So this is the protein that we're looking at, 427 kilodaltons. It interacts with actin, and this actin filament, again, will also interact with myosin. We don't see myosin here. And you can see that there are a number of repeating or very similar domains that we have here as well. The other thing that I wanted you to be aware of on this particular diagram, and we're not going to go into a lot of detail on that today, we'll cover a little bit more of it just as a refresher next week, is that when you look at the muscle, and this is muscle, this is the muscle membrane, which we call the sarcolemma. This is where we get the generation of action potentials that cause the muscle to contract in the first place, that there's always a link with the extracellular matrix. We are not going to ask you, or I'm not going to ask you to know all of the details of the extracellular matrix interacting proteins. However, the most important one being that when we look at dystrophin and how it interacts with the extracellular matrix, there are dystroglycans that you have here that provide sort of that link between the dystrophin molecule, actin, and the extracellular matrix. Again, that's the only detail that I'd like you to make sure that you know. Some of these other points that I'll come back to on next Monday, I'll highlight again. But when you're going back and studying this weekend, I really am using this slide just to emphasize again what I've shown you on the bullet points on this slide, to give you an idea of how all of this is coming together. Again, on this particular slide here, you do not need to worry about all of the different types of deletions, etc. If you are looking at the top, the only things that I'm going to highlight for you is that actin binding region. This actin binding region in every form of muscular dystrophy we look at, including Duchenne's, is not affected. What are affected, though, which is actually of use to us, are the domains that you see here, these rod domains, with all these different repeating regions that I showed you on this slide here, these repeating rod domains. These are the ones that make some forms of dystrophin just a little bit shorter or not quite as complete. And we know that there are different types of deletions that can occur. So whether it is a large deletion, but it is a contraction of the full size, or we get a premature stop codon that actually gets introduced in Duchenne's, where we change the reading frame and introduce a stop codon prematurely, we are affecting dystrophin. Dystrophin is not able to make those complete links anymore with the extracellular matrix. It can't really combine well with those dystroglycans that I showed you earlier. So this dystrophin anchors this molecule with actin and that protein complex that I showed you earlier. And every time we don't have dystrophin, so it starts off, we have it, there's a developmental isoform, because some of you might be thinking, why does the young male start off with a fully intact skeletal muscle heart system, and then what happens to cause this to occur? There is a developmental isoform that is not affected in Duchenne's, where they have something that compensates for this. This is lost in development, and then they start using dystrophin. And when this dystrophin is no longer there in sufficient quantity during development, then we have these different issues where we actually, when the muscles contract, the inappropriate form of dystrophin is no longer able to make a good connection between actin and the extracellular matrix. Every time that muscle contracts, that muscle is going to be damaged, and the muscle fibers are going to die through a process that resembles necrosis. And eventually these fibers will start to die off, which is why the individual starts off normally but then has developmental issues. Again, this is very, very controversial. I'm going to put this up here now, and I'm going to come back to it next Monday. But again, don't worry about all the details here. This is that complex that I showed you earlier. And this complex and the appropriate complex where we have this large dystrophin molecule making the appropriate connections with the actin as well as the extracellular matrix through these dystroglycans also seems to allow for the appropriate level of calcium balance. This calcium homeostasis that's so important in muscle contraction seems to be affected by having this complex normally present. One of the molecules that we'll come back to next week might potentially be this nitric oxide. I sort of skipped through that earlier. If you go back and review, you'll see that one of the domains on dystrophin is actually a nitric oxide binding domain, and this might be the link, but it is very controversial. We think that there is a loss of calcium homeostasis when we don't have dystrophin around. That complex is gone, and this complex being lost leads to an abnormal form of calcium during muscle contraction that eventually causes necrosis of those muscle fibers. Again, very controversial model. This is not the only model. We'll come back and revisit it in a higher level of detail next Monday. For the time being, I just wanted you to be aware of one potential contentious model system within this system. So in this lecture series, we're always going to be looking at a particular model. Ideally, as you'll think about later on in this course, if you can grow a mini organoid from someone's skin tissue, so I take a sample of skin fibroblasts, I clean them up, and then I can grow sort of a mini organ, whether it's a brain or something else, there is no reason why we couldn't grow mini muscle organoids. So again, keeping that in mind, the way that things are changing very quickly in science, up until the development of organoids, people wanted to take muscle biopsies. Have any of you ever had a biopsy before? Someone took a little chunk of your tissue out. Was it a pleasant experience?
It was an endoscopy too, so I
Endoscopy. Okay, so if you've ever had like a little biopsy where someone sticks in a long biopsy needle and removes a part of your tissue, it's not a very pleasant experience. It's not painful because they use local anesthetics, but it is very daunting to actually know that they're taking a little piece of your living tissue away. And so if you're asked to provide a number of these different muscle tissues, especially in a young developing infant, it's really unethical to do that. And until we develop appropriate sort of organoid models for skeletal muscle, this is the best way to actually study how the muscles are dying. We know that there are imaging studies that are fairly non-invasive, but they don't give us any molecular information. They can tell us that yes, as this individual with this genetic disorder, as they use their muscles, we see a loss of muscle fibers. That's really the best that molecular or medical imaging can actually do for us through MRIs, through 3D ultrasounds, etc. We're not really able to look at the molecular details to try to reverse them right now. So we often come up with these different model systems. So one of the most popular types of model systems are these dystrophin deficient models, including the golden retriever muscular dystrophy model. So it's a dog model, as well as the Delta E50 MD beagle, which is Eric Olson's new paper that I'll show you next week. So these are, again, model systems that very closely resemble the human form of muscular dystrophy. So we talked about the good reasons to use dogs in research. What are some of the not so good reasons to use dogs in research? Yes? It's unethical to use dogs?
As an animal lover, probably.
Yeah, as an animal lover, sure. Absolutely. Anything else? Yes?
They're harder to work with.
They are much bigger, they are more costly, for sure. So mice, you can put 3 or 4 of them in one cage, and they're fairly easy to take care of, but dogs are bigger and so more costly to take care of. Yes?
I imagine there will be greater genetic differences.
There could potentially be more genetic differences, which is why this Delta E50MD model was so exciting, because that does resemble the human situation. Yes?
There could be more regulations or
So definitely in terms of animal ethics research protocols, there are more regulations. It's harder to actually get an ethics approval. Yes, Leanne.
It also takes much longer to grow a model than a mice model just because they have a longer life expectancy.
Yeah, and because they do have a longer life expectancy, it does work for us because we can see, like, if this therapy is effective, we can actually track it for longer periods, but it also takes longer for the disease to progress. So all really good reasons. So there are always pros and cons to each of these model systems that we've been looking at. And for some of you who are really interested in this, the CRISPR-Cas9 system has actually been used to develop and knock out dystrophin in a rat system. And so by knocking it out acutely using CRISPR-Cas9, it's actually been another way and another tool for people to go back and generate new model systems related to Duchenne's muscular dystrophy. And again, it affects dogs very much, and these are, again, I'm not sure if you are aware of this. How many of you have dogs? Some of you do. I have a dog as well. And dogs have been undergoing breeding for a long time, which means that their genetics are have been affected for a very long time. And for the golden retriever model for muscular dystrophy, it's a spontaneous mutation that's developed. And unfortunately for this golden retriever, its hind legs are also affected. The same with the beagle on the right. The inbreeding of dogs has led to a number of really interesting neurological and muscular changes, including like dogs that have spontaneous epilepsy, dogs that have epilepsy when you flash bright lights at them, etc., mainly because of the breeding that were done to make the dogs really cute or small or really large, etc. So the one that we're going to start off with on Monday is this very well-established model system, which is called the MDX mouse model. And the MDX, which is the muscular dystrophy mouse model system, is probably the best sort of study. And this is where we have a premature stop codon, just like we see in that frameshift mutation in the human Duchenne's muscular dystrophy. And in the MDX, we also have exactly the same premature stop codon. So we also have a truncated form of dystrophin, not necessarily in the X chromosome, here in exon 23 in the dystrophin gene, but very similar to what we would see in the human. And again, this is a C57 mouse, which is a very commonly used form of mouse in both biology as well as in medicine. And one of the things that we see is that if you have a mouse where we have the mouse moving around, this mechanical damage, just as we would see in early development, would also result in the loss of these functional myofibers. And these are also looking very much like if you overuse a muscle and dystrophin isn't there, that you will have sarcolemmal tearing and loss of muscle fibers. And this loss will eventually cause the inappropriate calcium homeostasis, and that will disappear, and this will lead to myofibril death. The other thing about MDX mice, and again, I just use this to kind of gauge everyone's background, is that in these mice, they will also have an elevated level of NF-kappa B. So if I were to look at NF-kappa B in general, if I see an elevation in NF-kappa B, what do you normally associate that with? What is the pathway that you would normally associate it with? Yes?
Inflammation.
So this is normally inflammation. Agree? Disagree? So this is normally associated with something being activated in the inflammatory pathway. Not surprising, because we see that in human situations as well, where inflammation seems to go up along with muscle tearing. The other thing to make note of, and I talked about this very, very briefly, there is another molecule that kind of looks like dystrophin. This molecule is known as utrophin. And utrophin has about 80% homology and structural similarity with dystrophin. In fact, it has the actin binding domain. It has many of those rod coil domains that I showed you earlier with. And one of the things that unfortunately happens with the mouse model, when you look at the MDX mouse model, they lose dystrophin, it's true, but they don't look so bad. The mice don't look like the beagle model or the golden retriever. And one of the reasons why is that when people went back and examined it, the utrophin, which normally isn't upregulated, with the loss of dystrophin, this compensates for it. So this is, again, upregulated in the MDX model. We have to account for that or compensate for that. We don't see that in humans. And if you're thinking, why don't we try that in humans? People have, but it doesn't quite work as well in the human situation as it does in mice. And again, these MDX mice don't really look like the model system that we want because they have a normal lifespan. They don't die early. They don't have a huge dysfunction. They eat normally, they can actually still run around, etc. So a lot of that might be accounted for through this utrophin. Again, this is just to show you the homology. Please don't memorize this diagram. So when you're looking at these types of diagrams, I'll often tell you what you need to know. So when we're looking at this actin binding domain, it has the actin binding domain, so does utrophin. And if we're looking at the overall structure, it looks very, very similar. It has about 80% homology. It's just a shorter version of it. And in mice, especially MDX mice, when we lose dystrophin because of a premature stop codon, this upregulates and it compensates for this loss. Which means that MDX by itself is probably not a great model system to use. And again, outside of what I just showed you, please don't memorize this entire diagram. It's just to give us some context. And so the upgraded MDX model systems often take this into account, that when you lose dystrophin in these mice, and only in mice, not in humans, we have an upregulation of an 80% homologous protein known as utrophin. And one of the things that they do is to upregulate muscle loss through having the mice undergo forced exercise. Like they will be on a treadmill and they can't get off the treadmill, forcing them to exercise. How would you know if you're damaging the muscle? What would you look for? Based on today's lecture, if you have a really good form of short-term memory, you should be able to tell me. Yes? So I could look for CPK-MM. Anything else I could look for potentially? Yes?
Low levels of dystrophin?
I could do some muscle biopsies and look for lower levels of dystrophin. Yes? I could also look for NF-kappa B, all things that we talked about earlier. So again, those are the ways I like to ask questions. So again, maybe think a little bit about how you might go back and address that. So there are a number of these varying protocols which allow for different amounts of damage to muscle. And then there's also an MDX52 knockout mouse. So in this mouse, all forms, all homologous forms, short forms, truncated forms, everything are completely deleted. In this mouse, muscular dystrophy is actually very, very serious and very prominent. And so all revertant forms, including utrophin, are actually eliminated in the MDX52 model system. Is there anything else that you might come up with as a model system for muscular dystrophy? Anything that you can think of off the top of your head? Any ideas? Yeah. Paul? Could also try knocking out utrophin. Anything else that you could think of, potentially? So if you want to make something look a little bit like muscular dystrophy, what else might you target? Yes?
The calcium signaling pathway?
Sure. Controversial, but also I would take that sort of as a potential answer. So somehow upregulate the calcium signaling. And what's sort of the link between dystrophin and the extracellular matrix? Okay, so dystroglycans might be another model system that you can come up with as well. So again, I want to put this in some context for you. And this goes back to what I was showing you earlier. So Eric Olson, the last author on this paper here at the bottom. So he had a new paper that has come out in the last 10 days, actually re-examining it in a canine model. He did exactly the same thing in a mouse model system, also published in Science. If you can publish a paper in Science, you are a pretty good researcher. And I can tell you that we have a lot of research here at U of T, but being able to publish in a high quality journal like Science is actually incredibly difficult. So the fact that he took almost the same research and was able to publish it again in Science, the same journal, actually says a lot about the paper that he has recently published. But this is sort of the cool thing about CRISPR, and this goes all the way back to 3 years ago. For some of you, it might have been when you're starting U of T, or for some of you, it might be when you were thinking about coming into U of T in that context. When CRISPR-Cas9 started to make its rounds, and when people thought it was actually going to be a really popular therapeutic technique, there was this massive race on to be the first to publish it in a disorder where they could go back in and correct it. And one of those things, as I mentioned to you, was muscular dystrophy. And so at the very end of 2015, going into 2016, on the very last possible day of the year, in fact it was December 31st of 2015, in that one issue of Science, there was not just a back-to-back paper, there was a back-to-back-to-back paper. 3 different labs had simultaneously come up with the same discovery. That using this technique known as CRISPR-Cas9, that was just in its infancy, people were able to fix or correct this really serious disorder that we've just spent the last sort of hour talking about, muscular dystrophy. And so this was a dramatic sort of statement that this is going to be the future of what corrective therapy and genomic approaches to disorders, and we'll see this several times in this course, potentially could be. And what I'm hoping that you're going to do over the next little while is to think a little bit about this. We have a number of different genetic conditions where we have, through a mutation, introduced a premature stop codon, often leading to some kind of condition, whether it's something related to cystic fibrosis, muscular dystrophy, whether it is something that is a neurodevelopmental delay, or other types of very serious disorders. These premature stop codons are really interesting for us. And in the next sort of lecture, going through Eric Olson's paper and CRISPR-Cas9, we're going to be looking at this. People have been using drugs like adalurin, and this is a biotechnology-driven drug, which has been thought to actually allow for the ribosome to skip past. It's called exon skipping. There's a little premature stop codon. No problem. We'll give you this drug, and wherever there's a premature stop codon, we'll skip over that, allowing you to make a full protein. That has not turned out to be true. In fact, that drug, adalurin, is now thought to be somewhat fictitious. People don't think it actually works, although there are papers that were published on it. So we'll come back and talk about this in the next lecture, and why people are actually looking at muscular dystrophy and other disorders that involve stop codons as a way to develop therapies to be able to fix this. So if you're okay with this, in the next week or so, this is one of the things I'd like you to try to do. I'm going to open up discussion boards starting either later tonight or early tomorrow. If you have a potential topic and you're looking for group members, or if you already know the group that you want to work with, just indicate that on the discussion board. I'm looking for group members to help me with a Parkinson's paper, or I'm working on the genetics of Alzheimer's disease, or I want to use CRISPR-Cas9 and here's a potential paper. I'm looking for partners. Post something on the discussion board. In addition, I will be posting, with your help, a study guide. So from today's lecture, before that study guide goes up, can you think of like what lecture slides are important, not so important? Because in about 3 weeks' time, we have our first term test. To be honest, I would really like the average for that first term test to be, should we go for 80? Should we go for 85? Should we go for 90? No, let's not go there. I would love it, but that would be kind of dangerous for me. So let's just try somewhere between the class average being somewhere between 80 to 85. That would be awesome. The only way you can get there is to put in the time and effort. I'd like you to all do it. I will also, if we get enough discussions going, I will also be posting for you last year's term test, so you can get an idea of what last year's term test looked like. And we'll use one of the upcoming lectures to kind of go through the answers in class. So, Other than that, have a dry sort of Monday. Good luck with the rest of your classes. I'll see you all back in class next Monday morning. Okay. I think there was another student who said that she would be able to as well. So for this class, to be honest, the study guide is really straightforward. It's more of